always lovely to get an award, but as I'm sure everybody in my position would say, it's not ever one person who does research. It's a very collaborative enterprise, so this is nice to be recognised for myopia, I think. I think myopia research is moving into new areas now, looking at treatment, as we'll hear from Mark Bullimore tomorrow lunchtime. But again, following on from the last talk by Professor Koshi, myopia is a multifactorial disease with lots and lots of areas of research where I think we need to pull this together. So it's nice to have an award to show that myopia research, I think, is really moving somewhere, hopefully clinically more relevant than perhaps in the past it's been in the doldrums a little bit. The other thing I'd say is the college has been very brave to give an award for my own research, which is very fundamental, basic, some way away from clinical application. But I can do my best in this lecture to say why I think we need to do this ground level research in order to bring forward new treatments which will perhaps hit the clinic only in 20, 30, 40 years' time. Could I have my slides up, please? Oh, sorry. So I'm going to talk about this idea of a paradox of myopia in terms of the relevance of genes and environment. And they're going to talk about the latest developments in myopia genetics, and again talk more generally about myopia research in terms of what I think is good and also what is not so good, and where perhaps we can bring strands of research together to make ground-changing improvements. Now, hopefully this will be relevant to practicing optometrists in terms of, again, as Mark Bullimore will mention tomorrow, in terms of treatment for patients, which I think we can now start thinking about seriously, and in terms of genetic prediction of at-risk patients, and the idea, again, similar to the AMD story, how we can think about prevention as well. Before I kick off, I do need to cover a few important definitions, and those of you who, like me, are a big fan of this Radio 4, I'm sorry, I haven't a clue show, will be well aware that our vocabulary, our definitions are constantly changing over time, and even in the optometric world, words are taking on new meanings, and I think it's important we all try and stay up to date with this. So I'll run through a few of these definitions that I've come across recently and hope you might find useful. So I'll give you the word and then the up-to-date definition. So firstly, angioid streaks. Beware of accompanying Anne to sporting events. BIO, must remember to pay for that honey. Refract, umpire looks guilty. Direct ophthalmoscope, beware lending your instruments to a Welshman. Divergin, Welsh bachelor. Again, there's a Welsh theme here because of all my time in Cardiff. Dilation, happy Welshman. And of course, direction, aroused Welshman. So having got those things, we need to keep those in mind, obviously. There's been this big rise in the prevalence of myopia in Asia, and it's very concerning that 80% or so of school leavers in urban places like Singapore, Taiwan, Hong Kong are leaving school myopic or highly myopic, and in Europe, US, there's some evidence that prevalences are going up, perhaps not to the same extent. And so I think it's vital to find out what is driving this change. Is it genes, is it environment? And maybe by learning more about that, we can tailor treatments more effectively. We know there's a strong genetic element to myopia, and if you read the research literature on twins, on myopia genetics, you could easily get the impression that refractive error is wholly determined by genes. And indeed, if you look at this, the largest twin study that's been published with 4,500 pairs of twins by Chris Hammond in London, Dave Mackey in Australia, you can see that for the MZ monozygotic or identical twins, refractive error of twin 1 and twin 2 is usually very, very, very similar, again implying that genetics is really regulating how the eye develops its optical component parts and puts everything together, whereas in DZ or dizygotic twins, these are only as genetically similar as normal siblings, and you can see there's much more spread in that graph between the twins of a pair. Similarly, if you look at particular families, there seem to be occasions where myopia, particularly high myopia in fact, can be caused by a single gene defect, i.e. a mutation in a crucial gene somehow makes the eye become myopic either at birth or during childhood, and so you could follow the segregation or co-segregation of the phenotype, the high myopia, and the inheritance of a mutation. Again, implying that genetics is all important in that family. Then again, if you read a different part of the literature, you could get the impression that myopia is wholly environmental. And indeed, the increased prevalence in parts of Asia, that's happened over 1 or 2 generations, far too fast to be due to a change in gene pools, implying that it must be a change in environment or lifestyle. And looking at time outdoors and near work, we know that they certainly have an impact on the likelihood of a child being myopic. Here's some work carried out with Kathy Williams from Bristol, where when children were about 7 or 8 years old, their parents filled in a questionnaire saying how much time the child spent outdoors. And so we categorized the children as either spending a high or low time outdoors. And similarly, the parents reported how much time the child spent reading. Did they enjoy reading a lot, or did they not do much reading? And you can see from the separation of the 2 curves on each of the graphs that these predictors at age about 8 have a long-standing impact on whether a child's likely to become myopic then over the subsequent 7 years or so. So not a huge effect, certainly not the only factor that's contributing to whether children become myopic, but it does seem to be predictive, this environmental question when the children are young. Another indication that myopia is very connected somehow to the visual environment is the condition called form deprivation myopia, and this can happen to infants that are born with congenital ptosis, so the eyelid drooping down prevents them getting clear vision at any stage, or in the laboratory, researchers use this paradigm by deliberately fitting a frosted lens over the eye of an animal so that the eye can't receive clear vision, and that in all cases seems to produce quite high degrees of myopia. So evidence that the visual environment is the all-important factor in causing myopia. But can these factors both be all-important? Surely that doesn't make any sense. So this is what I think can seem paradoxical at times. I think there are a number of reasons for this apparent paradox. One is that, as I said, researchers tend to choose a study design that suits their idea of what might be important beforehand. So if you study twins, you've probably already decided that myopia might have a genetic component. So depending on which type of study researchers choose to do, can affect the outcome they can find. So if you do a twin studies make an assumption which prevents you ever finding an environmental contribution. Or if you do a form deprivation experiment, you generally just ignore genetics altogether and concentrate on this environmental stimulus. So perhaps some of the paradox is because of the way researchers work. The other thing is that if you're doing a study like an epidemiology experiment, you need to have variation in your risk factor in order to find an association. So in Hong Kong, Singapore, children don't spend much time outdoors. It doesn't matter who the children are, which social class, for example. If there's not much variation in that amount of time spent outdoors, if you do the epidemiology and you look at that as a risk factor, you don't find much association because there's not much variation there to look at the effect of. Whereas if you then try to look maybe at Singapore versus UK, where time outdoors would be higher in the UK compared to Singapore, you then have the problem that not only is it time outdoors different between the two countries, but diet is different, everything else about the society and lifestyle is different. So it makes it very difficult to pin down whether particular risk factors are important or just confounded with other variables that haven't been measured. But the work I'll talk about shortly is another potential explanation, is that genes and environment shouldn't be considered singly, but act together to contribute to risk. We'll come back to that. So I'd like to argue that both genes and environment can be important at the same time, but perhaps in the past we haven't done enough as researchers to consider things in a holistic manner. In terms of the implications for clinical treatment to prevent the development or slow the development of myopia, I think this might have some relevance. It's a little bit like applying the same drug to every patient. We know now with pharmacogenetics we can tailor the correct choice of drug to a particular cancer patient, for example. Depending on their type of cancer, one particular type of drug might be more effective, another type less effective. In the trials going on for slowing down myopia, we really completely ignore genetics altogether, for example. We really ignore most other things about the patients. We just give them treatment A or placebo, and then we look at the results. So that may be partly why the treatments have been a little bit disappointing compared to the animal studies, where everything in the animals' lives is rigorously regulated and controlled. Children are going to live very diverse, different lifestyles. Some of this could be the result of these disappointing results so far. Turning now to pure genetics, then historically over the last 10 years, which genetic research has come a long way, I think. If you wanted to study a condition, you thought it was genetic, you had to go out and recruit, find pedigrees, and so we did this in Cardiff for a long time with the family study of myopia, and so I know there are some researchers here in the audience who helped with that project and practitioners who very kindly gave up time and effort to recruit families for us, and so I'm very, very grateful for all that help. And we did learn quite a lot. We looked in single families. We tried to identify those that had a simple pattern of inheritance. Were they recessive, dominant, X-linked? And if we could find such families, then we can use well-powered methods to pin down a causative gene. Unfortunately, we realized, as did other groups around the world doing the same thing, that myopia doesn't often seem to follow this simple, recessive, dominant, it's called a Mendelian transmission pattern. Instead, it seems to be more higgledy-piggledy, much more complicated. So researchers thought, well, the solution now then, if we think myopia isn't just caused by one mutation in one single gene, but probably by mutations in different genes acting together, we need to pull lots of families together. And so again, we did this. We formed international consortia looking at hundreds of families at the same time. And again, we made a little bit of progress, but nothing very much. All we came away with the idea that myopia is always more complicated than we expected. Instead of maybe a handful of genes being important, this was telling us that it was probably tens or even hundreds of genes that are important in high myopia. with real considerable advances in DNA sequencing speed and throughput, it's now possible to sequence the human genome within a matter of hours and for perhaps £10,000. Or if you focus on the part of the human genome that codes for proteins, which is called the exome, then you can sequence that even more quickly and for just £2,000 or £3,000. And with your exome sequence, you can identify any mutation that isn't there in the database of other people's whose genomes or exomes have been sequenced. So this has revolutionized the analysis of single pedigrees. If you have a Mendelian disease, like retinitis pigmentosa or Stargardt disease, Salisbury's dystrophy, then with this exome sequencing technique, you can pinpoint mutations now very, very routinely. Unfortunately, this has been useful for high myopia, but again, because high myopia doesn't tend to show this single gene transmission pattern, it hasn't been as useful as we hope, but again, it's gradually leading to a trickle of disease genes which, when they're not functional, will give you high myopia. But we know now that they're only accounting for a handful of cases of the condition that we see in our patients, and that much more likely myopia is multifactorial, caused by interactions of many, many, many genes. You'll excuse the height, but I think it's fair to say that this year, and in fact the past month of February, myopia genetics has gone on a huge, huge leap forward. There have been 2 research papers that have taken the number of known myopia-susceptibility genes from 1 or 2 into the 20s or 30s. So this has been a step change in our understanding of what we know about the genetic cause, genetic susceptibility of myopia. So the first paper to come out was from a consortium of researchers around the world, including myself and researchers at Bristol, London, you name it. More than 100 researchers contributed together, studied 45,000 patients, so including, for example, patients involved in the ARID study. All these groups of patients were recruited together, pulled together, and we looked at positions on the genome that are related to refractive error in some way. The way we do this is we look at each chromosome in turn. So chromosomes are just a linear stretch of DNA, so you can go from one end of the chromosome to another. We know, thanks to the Human Genome Project, that there are particular places along the genome where people tend to vary from one another in the sequence, so they have a single base difference, which is what makes us different from one another. And so we can step through each chromosome, look along the length of each chromosome, and we end up with a graph like this called a Manhattan plot, which looks like a block of skyscrapers. And this is, if you imagine all the chromosomes laid out head to tail, going from chromosome 1 in red at the one end, chromosome 2 in blue, chromosome 3 in purple, and so on. And then we're looking at these known genetic markers laid out systematically, looking at positions where people vary in DNA sequence, and we're at each of these points we do a statistical test to say, is there an association between the genotype and that genetic position and a patient's refractive error? And we're looking for this linear trend of an association. So we plot the p-value of the test on the y-axis, and genomic position on the x-axis, and we end up with, we normally screen about 2.5 million genetic variants, so we end up with a 2.5 million points in our graph, and because we're doing 2.5 million statistical tests, our chance of getting a p-value less than 0.05 happens all the time, just by chance. So we have to set a very, very stringent p-value threshold to declare significance. So we normally have to get to a p-value of something like 5 times 10 to the minus 8. So a lot of zeros before we get excited. This is the outcome of the, it's called the CREAM consortium, the Consortium for Refractive Error and Myopia. So with these 45,000 patients, we're able to get enough statistical power to find some significant associations, and you can see highlighted these little blips where we get a significant hit for particular genes. So did you know, for example, the left-handmost one there, CD55 gene, I'd never heard of until we finished this study, is a myopia-susceptibility gene. CD55, if you go and look it up in the databases, it's an antigen for blood cells, not even sure it's expressed in the eye, and yet somehow that seems to be important in determining, I'll say important, have some influence in determining whether a child develops myopia. So the nice thing about genetics, from my point of view, is that you don't have to make any assumptions. It just churns out the genes that have a role. You don't have to decide, is it going to be a scleral gene, is it a retinal gene? This is what it is, and then you have to go and work out how it could be having an effect. So some of the results do make some sense. There's a gene there called LAMA2, LAMA2. That's an extracellular matrix gene from the sclera that we can piece together from its known functions that it might regulate how scleral fibrils are held together tightly, and maybe if this gene has a slightly different sequence, slightly different expression level, it may influence how scleral collagen fibrils slide over each other, for example. Some of the genes do make sense like that. Others are completely unexpected. For example, there are some ion channel genes that control potassium and calcium flows within the retina. So maybe the way the retina sees and responds to light somehow leads to some children being more susceptible to myopia. So we've now got 20 or 30 new leads, each of which is telling us how myopia susceptibility arises. But again, it's complicated. Why is CD55, for example, involved in myopia? It's got something to do with blood. It shouldn't have anything to do with myopia, but it does. So we need to go and do more research, find out which pathways are involved, and the big hope, because of the complexity, and as Dr. Koshi said a moment ago for AMD, the complexity means that gene therapy isn't an easy solution as we once hoped. But if we can find pathways through which these genes act, find underlying mechanisms, then we perhaps have better chance of finding drug targets. With either gene therapy or regular medicines, we can change the level of activity of these pathways to hopefully slow down myopia progression. The second paper I mentioned, that also came out in February this year, used a slightly different approach. It's from a single group of researchers, and they're not even academics. They're based at a company in the United States called 23andMe, and it's a company that does genetic testing. So if you go to their website, you can have a go. You can send them a saliva sample. They'll extract the DNA from your saliva, a bit like we used to do with our family studies, and then they'll look at all the genetic variants in your DNA, and they'll send you a report saying which diseases you're at high or low risk of, and how you might like to change your lifestyle accordingly. So they also looked at around about 45,000 participants who'd gone onto their website, sent them a saliva sample and said, yes, we're happy to help in research. And they looked at age of onset of myopia as their phenotype, but the results were surprisingly similar to the CREAM consortium's findings. So again, around about 20 or 30 genes, many overlapping hits. And the overlap is good. It shows that the findings are probably not false positives. We have to be very careful doing all these multiple tests. We're always wary that we're getting false positive gene hits, but the fact that the two independent studies came up with similar results gives us a lot of confidence. Another thing it showed is that for the researchers based in Singapore, Taiwan, Hong Kong, the genes that they were discovering and the genes that were found in Europeans were overlapping on the whole. So there didn't seem to be a difference in the susceptibility in terms of ethnicity. So whether that will hold up as we discover more genes, but at the moment we're not finding a cluster of genes, for example, that lead to myopia in Asians and a different cluster in Europeans. At the moment, the overlap is looking good. So back to 23andMe, they can tell you, for example, whether your risk of type 2 diabetes is from 8% to 52%. I don't know about you. Would you like to know this, or would you prefer to stay in ignorance? I'm not sure about myself. But thinking about the same situation in myopia, are we now at a stage with this cluster of 20 or 30 new susceptibility genes? Can we start predicting whether particular children are at risk? I took a look at this in the Children of the 90s study. So I've plotted on the graph here a genetic risk profile. So on the x-axis, you're going from the right-hand side where you have a very low genetic risk profile, coming up to the left where you have a very high genetic risk profile of being short-sighted. Because there are more hypermetropes than myopes in the sample, the blue bars in general are higher than the green myopes. And you can see that at the -4 end, so the left-hand end, there's not much ability to discriminate people who will or will not become myopic. But on the right-hand side, there's quite good discriminating power about who will and will not become myopic if you have a very low risk burden. Unfortunately, what this graph hides is that the underlying frequency distribution is bell-shaped. So this right-hand tail, where we have good predictive power, is only corresponding to a very small subset of the population. This gives a very rosy picture. Indeed, if we take the whole population, then even our 20 or 30 new myopia susceptibility genes only have the ability to predict about 2 to 3% of the variability that we see in refractive error within the population. So we're a long, long way from finding the genetic cause of myopia. We've really just got the tip of the iceberg in our 20 or 30 people. There are probably hundreds or thousands more that again have a weaker and weaker effect, but the sheer number of them is what we're missing. So going back to our Manhattan plot for refractive error, the genes that we're not detecting are those that are in the middle of the graph. So they're not high enough on our peak to say, yes, they're statistically significant. So they may have a p-value of 0.001. Because we do multiple testing, we can't be sure whether that's a true or false negative. So the other genes that we're not able to say for sure are probably that cluster in the middle of the graph, where we just need more and more and more patients, and we'll have more and more statistical power, and then we can start to identify even more genes. And hopefully the more genes you have, you know about, the more you can see how they're connected into physiological pathways. So that's where the work is continuing to head. Going back to risk prediction, this is a paper, again, hot off the press, looking not at myopia but for 7 other common diseases, going from hypertension, cardiovascular disease, type 2 diabetes, bipolar disorder, Crohn's disease, rheumatoid arthritis, and type 1 diabetes. And it's on the y-axis, it's plotting the area under the curve for a discriminating test. So if you plot your sensitivity and 1 minus specificity graph and get your ROC curve, this is the area under curve, so how much screening power you have effectively. So for some diseases like type 2 diabetes, the area under the curve is only 60%, so not much better than the 50% chance level, whereas for type 1 diabetes, you have about a 90% predictive capacity. So depending on the disease and its underlying genetic architecture, your ability to genetically predict who's at risk can be high or low. So thinking back to the last lecture on AMD, where although there are many, many, many genes involved, there's a small handful of AMD genes which seem to be very, very predictive of risk. So the AMD level might be quite high up on this chart. Unfortunately, myopia at the moment is going to be pretty low down, with our 20 or 30 known genes and a few more we think might be involved. Again, unfortunately, we're not there yet, but I think in the medium term, we may be able to, with a simple genetic test, which is becoming quicker and more easily interpreted with time, maybe we'll start predicting children who are at higher than average risk. Or perhaps again, coming back to the idea of tailoring treatment, we might be able to find children who have a genetic profile that makes treatment A a better option for that child than treatment B, so that be ortho K or multifocal lenses, whatever. But at present we do a very poor job, except for this small group of patients who have a very low genetic risk burden. You can be pretty confident that they won't go on to develop myopia. Quickly looking at current research projects, so we know there are clinical trials going on for treatments designed to slow myopia progression. My argument is that we need to do these studies because that's going to inform clinical decision making today and tomorrow, but we need the basic research, the fundamental research as well, in order to develop new treatments that hopefully will be better than the current generation, where we're getting about 30, 40, 50% success rates. So there's ongoing research into genes, looking at, again, families, twins, animal models, and environment, again looking at animal models of how the visual environment triggers myopia development. People following cohorts of children, doing epidemiology studies, trying to find associations between diet, lifestyle, etc., etc. But what I think is lacking is this holistic approach of looking at genes and environment together. So I'd like to finish by just talking about some research we did, probably one of the first attempts to try and put the two ideas together. So the question comes back to this idea of, is the epidemic of myopia in East Asia genetic or environmental? The ideal experiment to test that theory would be to take some children away from their parents, raise them in a different society. So we could take some children from Singapore parents, take them away, put them in the UK and see whether they become myopic, take some children from UK, raise them in Singapore and do likewise. But of course that's ethically not going to be allowed, nobody's going to let me do that. So because we can't do those ideal, well-controlled studies in children, we have to, I think, sometimes turn to animal models. So for this study we used chickens, which have been used very widely in myopia research, and again it's this form deprivation paradigm where we cover one eye with a frosted lens, so the eye we cover becomes short-sighted, the other eye remains normal. And we started off with some genetically diverse chickens, about 200 of them, and we treated them all identically, and we checked how myopic they became. And because chickens have a real remarkable ability to emetropize, we only needed to study the chicks for 4 days. So they had 4 days of frosted lens wear, then we saw how myopic they became. And we kept those chickens that became very highly myopic, and we paired up males and females who both became very highly myopic. And then we also paired up separately males and female chicks who didn't become very myopic. And then we looked at the offspring from those parents to see if there's any difference in the susceptibility in the second generation. So the idea being that if this is a genetic susceptibility, we should start seeing some split, some divergence in susceptibility of offspring. So here's the scheme in a different way. The red bell-shaped curve is what we expected for the distribution of myopia after our treatment. So we give a uniform treatment, but we expected to find that some chicks are more susceptible than others. And then we take the high susceptibility chicks, so the blue color, and again form deprive them. And we did this for 2 generations, and you can see on the right, chicks start off being lovely, cute, fluffy, furry, cuddly animals, and within a few weeks they grow into monsters. So these chickens would grow, particularly the males, about 3 feet tall, become very, very aggressive. So I became the most unpopular researcher in Cardiff because none of the animal technicians would go near these chickens in case they got attacked. You go into the room, which had 20 chicken pens, and all the males would immediately start crocodiling, huge volume. So it was a very interesting experiment to perform. Anyway, here are the results. So we started off with our initially genetically diverse chickens, and just as we hoped, we got a roughly bell-shaped curve of distribution. So some chickens became -25 diopters myopic, some only -5. But then when we took the high and the low chicks, bred a subsequent generation, we started to see some split. Then the highly myopic line became a little bit more susceptible than the low susceptibility line, and in the second generation, that split became really surprisingly apparent. And I should mention that all the experiments were done masked, so we were refracting chicks, and they're all numbered, so we knew in our records which chick was in the high or low line, but when we were measuring them, we had no idea. So until we unmasked, after 2 years, we had no idea whether the experiment was working or not. But you can see that genetics seems to have a big role in this purely environmental form of myopia. This is the same data plotted on a family-wise basis, so each bar are offspring from a particular set of parents, and you can see from the high susceptibility parents, some families of chicks are all highly susceptible, and from the low line, some families of chicks are all very low susceptibility. So again, reinforcing the idea that it's a strong genetic element. And when we do the stats, we find that about 50% of the variability in this form deprivation environmental type of myopia can be explained by genetic background. We wondered whether this was a timing issue. Were the chicks that were not becoming myopic, were they just being delayed? That didn't seem to be the case, because if we continued the form deprivation for 10 days, then the difference between the two lines became even more striking. So it didn't seem to be just a lack of catch-up. And if we induce myopia by raising the chicks with a negative lens over one eye, which is a different kind of visual cue, but again, the minus lens will trigger myopia development as the chick emetropizes. We found a difference between the two lines in that response to negative lens wear, but when we fitted chicks with a positive lens, they became hyperopic to the same extent. So this implies that maybe emmetropization operates slightly differently depending on whether the eye is trying to grow faster or whether it's trying to slow down its rate of vitreous chamber elongation. So it seemed that when we look at genes and environment together, we can start to explain a little bit more about what might be going on. So certainly in chickens, we know that this purely environmental form of myopia is strongly genetic. It doesn't seem to be just a delayed reaction, and using different experimental treatments to induce myopia seem to share a common genetic pathway. So the hope would be that if we can find genes that are important in these chickens, maybe those same pathways could be being used in children developing myopia, even though children aren't wearing frosted lenses, they're not wearing negative lenses. Maybe if this is a common conserved pathway that all kinds of myopia are feeding through the same pathway, then maybe up or down regulating that pathway can help to slow down childhood myopia progression. So I think we're getting close to the stage now where genetics can do something useful. We're probably not there yet, but we're doing all this research. It's going to be a real shame if at some stage we don't start getting some payback. And I think the sorts of things we'll be able to help with are predicting children at high risk, tailoring treatments to particular patients, and again, maybe reassuring particular parents who think their children are at a risk can try to say something better than, well, if you have two parents who are myopic, the chance of a child being myopic is high. It'd be nice if finally we can go beyond that. I'd just like again to thank my collaborators, particularly John Erickson in Cardiff and Kathy Williams in Bristol. Thank you very much.
Very much, Jeremy. That was fascinating. It's great to see that we're getting somewhere with this. We might be taking saliva samples from our patients and sending them to labs. So, any questions for Jeremy? One from Adam, just in this instance, please.
Thanks. Hi there. Fascinating lecture. I can understand it's very interesting research as well you're doing. Two questions. One, how do you refract a chicken? And the other one being a bit of a devil's advocate in the way that why do you think that it's necessary to continue the myopia research into the underlying causes when we've got treatment for it, i.e. glasses, contact lenses, and it's not like AMD, for example. Endpoints like that.
The first question, we use retinoscopy to refract a chicken, and if the chicken is awake, it's difficult because chickens have a huge amount of accommodation, but if you anesthetize them and hold their eyes open with a lid retractor, then they don't accommodate while they're asleep. So it turns out to be a little bit like refracting a baby, I imagine. If you can, to me, refracting a chicken is much, much easier than refracting a baby. The second question, again, yes, we have treatments at the moment, but they're not as good as we'd like them to be in terms of slowing down myopia progression. And also there's a lovely article recently by Ian Flitcroft, who makes the point that although we think about only the very high degrees of myopia being the cause of pathology, and that if you're -10, -20 myopia, your risk of retinal detachment goes through the roof, but he argues that because myopia is this distribution where you have many, many people who are -3 and only a handful who are -10, because the risk of retinal detachment, for example, is higher for a -3 person than it is for an emmetrope, that risk, because of the number of myopes, is actually much greater and more clinically relevant than the -10s who in the past have all been worrying about. So he makes a really convincing case that in terms of the burden of disease on society, that the -1s, -2s because there are so many of them, and some of them do go on and have myopia-related pathology, either through connection with glaucoma, where myopia is a refractor, etc., etc., that there's a sheer burden of society in eye disease does warrant attention. And again, in Japan, for example, myopia, where prevalence is very high, myopia is now one of the 2 leading causes of blindness. It's catching up and may soon overtake AMD in Japan. And again, in other parts of Asia, that's going to happen sooner or later.
Dr. Guggenheim, interesting talk. I'm glad to see you didn't condemn all myopes as being in some way genetically malfunctioning. I've often felt that some forms of myopia, shall we say, are within the normal range. For example, I'm not a 6-foot Adonis, much as I would like to be. Does that make me abnormal? The second thing is, one time, a few years ago, the Institute of Aviation Institute of Medicine at Farnborough discovered empty field myopia, which has affected pilots flying very, very high. And they had nothing to fix on. Any comments?
Yeah, for the empty field myopia, that's definitely a phenomenon that in certain situations the eye will take on an accommodative state that makes the patient myopic. So again, that's something to be wary of when we're prescribing for patients who may be driving at night, for example, where there's perhaps not enough accommodative cue to give them a good accommodative focus in the distance. They may suffer blurred vision as well as the glare from the headlights. So I think empty field myopia is definitely a well-researched, well-recognized clinical condition. Typically, around 0.75 diopters myopia will be the usual level for that. Coming back to your idea of, again, I think you're getting at particular genetic forms of myopia. You're not a 6-foot Adonis, neither am I, but there certainly seem to be these rare forms of myopia that are caused by maybe single gene defects, and in that sense there are these extreme cases. Often they do have extreme levels of myopia, the -20s, -30s, -40s. Often these patients will have a particular very deleterious mutation in one of their genes. So I think there's a whole gamut from the idea of the general population, where we're thinking of hundreds, thousands, tens of thousands of genes, a bit like being a dealt a pack of cards, and depending on your hand, each of these genes, or variants of these genes, pushes or pulls you away from the emmetropic range, and if you're unlucky, you get a hand of lots and lots of myopia-susceptibility genes. You're pushed into the realm where, given your environment, you're very at a high risk of developing myopia. So I think there's a whole range.
Thank you.
If I can hark back to one of the graphs you showed us before about the relationship between the amount of time children spend outdoors against near vision, could you just remind me what age that started from? Because in my time in my own practice in northwest London, dealt a great deal with the Orthodox Jewish community, who encouraged their children to start studying at a very, very young age, and really don't encourage a great deal of outdoor activity. And it began to strike me that it possibly the age at which intense near vision is commenced that has as much effect as maybe even the genetic links with the parents.
That's a lovely question. The study we did looked at from the age of 7 to 15, and the time outdoors was measured when the children were about 8 on average. But if you look at countries around the world where myopia prevalence rates are very high, so Singapore, Hong Kong, Taiwan, the children are starting kindergarten when they're about 3. So to get into a school in Hong Kong, you need to to get into a primary school in Hong Kong, you need to have gone to a very reputable kindergarten. To get into a good kindergarten, you have to have awards for music and drama, and you have to be able to read and write. And so in Hong Kong, children, as soon as they're old enough to walk and speak, are being tutored at home, and then going through very high intensity kindergartens up to the age of 5, by which time they'll have developed their CV to such an extent they might get into one of the better primary schools, and from there they work their way up. If you get into a good primary school, you're likely to get into a good university. So in Hong Kong, it's been a tremendous eye-opener. The amount of effort parents put into educating their children it's just been staggering. And so I think you're absolutely right. The age children start near work could turn out to be absolutely crucial, I think. But it's very difficult to do a controlled experiment there, because if you look at Western families in Hong Kong, they need to get their children into school, so they again start tutoring at home, sending their children to kindergarten. So there's very little variation where we can find out whether that is the risk factor it may well turn out to be.
Thank you.
Just going back to the previous question, I think, so conditions such as Marfan's, where they're generally genetically inherited, and there's a very high risk of myopia in those types of conditions, I just wondered whether the genes involved in those kind of inherited conditions are linked with the ones that you've found.
Again, lovely question. The genes for those conditions are generally known. So Marfan's, for example, is a gene called fibrillin, which is a gene involved in elastic fibers that connect the zonules of Zinn. So the lens often dislocates in Marfan's, as you know, because of defects in that fibrillin gene. So far, there's been a little bit of overlap in the genes we've found between these syndromic conditions where myopia is a feature compared to non-syndromic, just regular school myopia. But the overlap, I think, is artificial. Because of conditions like Marfan's, researchers have done candidate gene studies where they've deliberately set out only to test genes like fibrillin, collagen, other genes important in these rare, purely genetic conditions. They've gone out and tested those in cases of case control studies where they've got high myopes versus emmetropes. And sometimes they show a small link, but when we do completely unbiased screens, like the CREAM study, the 23andMe study, we don't see the same genes cropping up. So as we do more and more studies, we may find they start to show up later on. We don't know yet, but at the moment, it doesn't seem to be the overlap. So I tend to view it that if you want to develop a normal eye and a normal refractive state, you need to have functioning genes. You need to have genes that generally perform the task they should do. You need to make the right protein. That protein needs to do its normal job. And so there are going to be a huge number of proteins involved in making a normal eye. If any of those go wrong, yes, you can get a rare high form of myopia. But I think in the general population, it's not an all or nothing condition where the gene doesn't work or a particular gene doesn't work. It's just all these variations of genes, all got different flavored genes, all of which are subtly different from each other. And in those situations, for example, the genes we're finding here are some of the last we would have ever expected. And we're finding, for example, one of the genes, when it's mutated, causes a hearing defect, not an eye defect. So again, we've got to take a step back and think, well, what is it that makes children susceptible to myopia? And take away all our preconceived ideas, I think, and then we'll perhaps make the fullest use of the new information we've got.
Okay. Take one last question from Deputy, please.
Just a curiosity, I work in a hospital where I do biometry for cataract patients, and I was very curious a few years ago. We did a 15-year-old male. He came up -0.5 afterwards, and then we saw him about 3 years later. He was -1.75. And the impact of what the lens, we removed the natural accommodating lens, has gone IOL. Have you looked at models of maybe age group of kids, I don't know, teenagers to 20, to see what happens if they've had cataracts done. Why do they develop myopia? So do we exclude the lens in this case, or is it something else?
Yeah, that's an interesting report. I've heard similar stories before, and it seems to be the case that the lens is somehow communicating with the retina and controlling normal homeostatic eye growth. So again, if you look at many tissues in the eye, they seem to be sending signals to each other, perhaps to control how the eye develops as a kind of coordinated whole. It's pretty remarkable that the lens gets just the right shape for the size of the eye, etc. And it seems to be that cells are sending signals, and so if you replace the regular crystalline lens and that signal is not being transmitted or not being picked up, again, that could be some of the reason why you're getting myopia in that particular patient. It's a bit of speculation.
One last thing. If you looked at Southeast Asian population, have you looked at the Swedes where the kids start school at 7? Do they have a different model of myopia?
Again, don't know of any epidemiology in Sweden, but in other Scandinavian countries which are also fairly laid back about levels of myopia, it seems to be on a par with UK. So that doesn't mean that early education is not important, but it could be there's a critical age. If you're starting school at 2, 3, 4, maybe that does have an impact, whereas if you're starting at 5, 6, 7, that difference between 5, 6, 7 is not so crucial perhaps. But
Okay, thank you very much, Jeremy. That's fabulous. Thank you.